Would you believe that inside and on your body there's a hidden world, teeming with life? Trillions of bacteria, archaea, fungi, and viruses occupy your gut, your skin, your mouth. These microbes are not just hitchhikers. They co-inhabit you, deeply influencing who you are. All of these organisms together make up what scientists call the human microbiome. The combined genetic content of that microbiome, the metagenome, vastly outnumbers the code in your own human genome in terms of gene diversity. While your genome is fixed, inherited, and relatively limited in how it can change, the microbiome is dynamic, shifting with diet, environment, medications, even your moods. It turns out that this metagenome may be one of the most important determinants of health, in some ways more influential than our human DNA. By the end of the video, you will fully understand how the human microbiome, its metagenome and dynamic functions, may have more immediate power over health and disease than your static DNA, why changing what lives inside you can alter disease risk and metabolic outcomes, and just how scientists are using cutting-edge techniques to measure, map, and manipulate that influence. To understand that, one must first grasp what researchers have discovered in recent years. The human genome, about 3.1 billion base pairs in diploid nuclear DNA, encodes roughly 20 to 25,000 protein-coding genes. It forms your blueprint, the basic architecture of your organs, your cellular machinery, innate predispositions to disease. Yet we share more than 99.9% of that genomic sequence with each other, so variation between individuals, in terms of predispositions and traits, is modest. This means that while there is some influence, many differences in health, disease risk, metabolism, immune function, behavior, even aging aren't fully explained by human DNA alone. Enter the microbiome. The gene catalog of microbial life within us contains millions of protein-coding sequences, many entirely unlike what we find in human genes. Advances in genome-resolved metagenomics, especially using long-read sequencing technologies and hybrid assemblies, have brought this into sharper focus. With metagenome-assembled genomes, MAGs, scientists can reconstruct nearly complete genomes of previously uncharacterized microbes. These reconstructions often reveal structural variants, mobile genetic elements, and plasmids, pieces of genetic material that move around, carry specialized functions such as antibiotic resistance or metabolite production. A recent review, Genome-Resolved Metagenomics, a game-changer for studying the human microbiome, 2024, Kim et al., makes clear how whole metagenome sequencing is enabling discoveries of novel microbial taxa, expanding functional gene catalogs, and opening new vistas for microbiome medicine. Metagenomics is not the only technique. Multi-omics, combining metagenomics, transcriptomics, proteomics, and metabolomics, now lets us not just list which genes are present, but see which ones are active, which proteins are produced, which metabolites are circulating, and how the host responds to them. A 2025 article advancing gut microbiome research, the shift from single omics to integrated multi-omics, Yang et al., illustrates this transformation. By layering measurements of gene expression, protein abundance, and metabolites, scientists are mapping how microbial activity translates into real physiological effects. One of the core findings across many studies is that core physiological functions depend on the microbiome in ways human DNA cannot replicate. For example, digestion of dietary fibers and complex nutrients, fermentation of food components, and production of short-chain fatty acids, SCFAs, like butyrate and acetate, are functions carried out by microbial enzymes. These SCFAs bind to host receptors such as GPR41, GPR43, or nuclear receptors like FXR, altering metabolic pathways, modulating immune responses, regulating inflammation. Synthesis of certain vitamins, especially B vitamins, and other essential metabolites occurs by microbes. Humans cannot produce some of these themselves. The immune system requires training. Microbial antigens and microbe-associated molecular patterns, MAMPs, help in establishing baseline immune tone, regulating Th17/Treg cell balance, IgA secretion, preventing overgrowth of pathogens, maintaining barrier integrity, tight junctions, mucus layer. These functions are repeatedly highlighted in recent papers exploring gut microbiome ecology, immune disease, metabolic disease. See Systematic Framework for Understanding the Microbiome in Human Health and Disease, Zekima et al., Signal Transduction, and Targeted Therapy, 2024. But what are the human genome's role? It remains vital. It sets predispositions, encodes cellular machinery, organ formation, inherited risk of many diseases. Yet variation between individual human genomes is small relative to what microbes bring. Most people share 99.9% of basic human genome sequence, so differences in phenotype, disease, metabolic capacity often depend on environmental factors and microbial interactions. The environment, diet, use of antibiotics, lifestyle choices, geography, these influence which microbial species take hold, how they function, and how their genes are expressed. Studies of heritability show that some taxa of microbiome composition are influenced by your genetics, but the majority of the variation arises from non-genetic sources. Twin and large cohort studies controlling for shared environment show that after controlling for such factors, heritability is modest. The ability to change the microbiome more rapidly than our human genome gives it great power. Interventions such as diet changes, prebiotics, probiotics, antibiotic exposure, and especially fecal microbiota transplantation, FMT, have shown measurable effects on metabolism, immune status, disease risk, sometimes even behavior. One clinical trial involving individuals with metabolic syndrome who underwent FMT from lean donors showed improvement in insulin sensitivity. In clinical effectiveness of fecal microbial transplantation for reducing insulin resistance and abdominal obesity, Wang et al., 2025, reported positive metabolic changes following FMT in those individuals. Another study, fecal microbiota transplantation, reverses insulin resistance in type 2 diabetes by Wu et al., demonstrated that in animal models, transplanting gut microbiota from healthy donors improved glucose metabolism and insulin tolerance. Meanwhile, a trial combining a Mediterranean diet with lean donor FMT in metabolic syndrome subjects showed some beneficial changes, including weight loss and improved lipid profiles, though not all metabolic endpoints responded strongly, and the effect sizes were variable, highlighting both the promise and complexity. These intervention studies also reveal heterogeneity. Not everyone responds the same. Baseline microbiome composition matters. Individuals whose gut microbiota allow good engraftment of donor microbes tend to have better metabolic responses. In recipient microbiome-related features predicting metabolic response to FMT therapy, Zhang et al., 2024, responders had higher engraftment rates of specific donor-derived microbes. Another recent review of FMT in type 2 diabetes emphasizes that while FMT can transiently improve insulin sensitivity, sustained effects are more reliably observed in patients whose microbiome signatures at baseline are favorable. Otherwise, changes may fade or be inconsistent. On the technical side, it is not enough just to observe associations. Scientists are increasingly using genome-resolved metagenomics to reconstruct MGs, utilizing long-read sequencing and hybrid assemblies to uncover plasmids, mobile elements, structural variants that were previously invisible. A substantial review in Nature magazine in 2024 described how these techniques yield far greater insight than traditional 16S rRNA gene surveys. Furthermore, the integration of multi-omics, combining data from metagenomics, which genes are present, transcriptomics, which genes are being transcribed, proteomics, which proteins appear, and metabolomics, which small molecules are produced, enables understanding of not only what microbes could theoretically do, but what they actually do in real time inside humans. For example, advancing gut microbiome research, Yang et al., 2025, describe pipelines that link microbial gene expression and metabolite production to immune or metabolic outcomes. Causality has been probed in notobiotic and germ-free animal models, where transplanting microbiota from donors with specific phenotypes, lean versus obese, high versus low inflammation, transfers metabolic traits. Those models show that microbes alone can cause changes in glucose metabolism, immune profiles, even the structure of inflammation. Such experiments, together with human FMT trials, provide proof that microbiome-mediated effects can be more rapid and modifiable than genomic predispositions. Despite all this promise, there are important limitations and nuances. Not every FMT or probiotic intervention works. Some clinical trials in type 2 diabetes have found only mild or transient improvements in glycemic control or insulin sensitivity, with many endpoints failing to reach statistical significance or sustain improvement after time. For example, a 2024 trial microbiota transplantation in individuals with type 2 diabetes in Spain found that FMT from lean donors along with probiotics showed changes in microbiome composition, but no significant improvement in insulin sensitivity or HbA1c or OGTT results over 12 weeks. Also, donor variability, baseline microbiome diversity, host factors, including lifestyle, diet, medication, genetics, all influence outcomes. Methodological challenges remain: controlling for contamination, dealing with low abundance taxa, increasing sample size and duration, achieving strain level resolution, standardizing sampling and bioinformatic pipelines. Moreover, most data so far comes from populations in higher-income countries, Western diets and lifestyles. Underrepresented populations may show different patterns. Genome-wide metagenomics datasets are still geographically biased. Putting this all together, the bottom line becomes clear. Human DNA gives us the stable architectural framework, the inherited predispositions, the structural and developmental baseline. But the microbiome is a dynamic, responsive, gene-rich system that interacts continually with our environment, modulates immune function, shapes metabolism, affects how we digest, how we respond to threats, how we heal. Because it is modifiable through diet, lifestyle, antibiotics, medical interventions like FMT, offers opportunities for change and improvement that human DNA cannot provide. For many health outcomes, insulin sensitivity, inflammation, disease risk, even how well we age, the microbiome may often be more immediately influential, more actionable, and thus more important than our static genome alone. In the coming years, as genome-resolved metagenomics, long-read sequencing, multi-omics integration, and precision microbiome therapeutics advance, we will increasingly see medicine that doesn't just read our genome, but actively modulates our metagenome. The promise is not to replace the significance of DNA, but to recognize that our health is governed by the intersection of our static blueprint and our dynamic microbial partners. It is here, in that intersection, where much of our future potential for prevention, therapy, and optimization lies.